Twitter. It's just like really frustrating to like kind of get out there, and also like my content on my blog is not really packaged all that well, which kind of sucks. So that's what I'm going to be kind of working on. I've got this amazing idea. I've been sitting on it for like a year. It's something that I wanted to write about, and oh no, they can't see those things. Those are top secrets. These are all the top secrets, guys. These are all the top secret things. Uh-oh. Oh no. Ah, it's not funny, because I cry myself to sleep at night. Anyways, don't look at the top secrets, guys. Did you know that, like, pi actually stops? Yeah, like, pi, after a while, it doesn't repeat, it stops. And the last digit of pi is a closely guarded military secret. They have it in Fort Knox, just the last digit is written down on a piece of paper. And they put it on Fort Knox with the gold. These are jokes that work really well if you're like doing stand-up at the NSA. They have stand-up. They have stand-up at the NSA, and they also have hookers apparently. Just so you know, job benefit, like stand-up comedians, but they do math jokes. It's not really that great, but they have hookers. I've never hookered before. I don't really plan on it, but they've got them if you want them. They will talk to you. Apparently, I don't know. I get like NSA.com stuff on my phones, and they're like trying to like hook up with me and bring me back to hotel rooms. It's weird. It's like, you went straight for the hotel room? Really? How desperate are you? This is gonna be fun. Somebody's got abandonment issues or something. This could be fun. Anyways, I will probably start. Probably gonna go stream, or not stream. I'm gonna stop streaming, and then I'll be back to write this excellent treatise. I think that's how you say it, because I never went to school. Sorry, Daddy, I didn't go to Oxford with your money. I went to go hang out with NSA hookers, and I got balloons of dope and partied with base nectar. I didn't go to Oxford, Daddy. Anyways. Oh, yes, changes in the cytochrome P450 from long-term drug usage. You can do this. It's really easy. You can do it. All you have to do is go to Walgreens and go on a trip to Walgreens and get some loratadine. It's an allergy medication. This is a field trip for the kids. 8 and under only. Never mind. Anyways, you can actually play around with like your own liver. You can play around with how your liver expresses its genes with allergy medication. Isn't that amazing? Like an allergy medication, like significantly, and like this allergy medicine, like very significantly alters how your genes are expressed in your liver. By the way, this medication right here, and this is programming. By the way, this is my blog's title is Metanomics and Probabilistic Programming. This is an intro. This is for part 607 of Class 2E. I've never been to college. I went to college once. I went to Virginia Tech, and I remember I met 3 people that I didn't already know from high school. 3 people. There's a problem. There's a very serious problem with that. I went to college. I paid them $25,000, and I wasn't exactly in the mood to like learn in 2005, but I went there. I paid them a lot of money, and I'm a little butthurt because I made 3 friends. Actually, that's not really. It's just an indication that something did not happen correctly when I went to college. Anyways, the point is, you can change how your liver expresses these genes. And one of those genes is called PXR, and PXR promotes a one of the cytochrome P450 uh, like genes. It promotes the gene. It promotes the enzyme that breaks down haldol, which is a common antipsychotic, and it's also an antipsychotic that's being used in implanted devices, similar to like birth control. Like now they have this like long-term like haldol like thing, and basically it's like the implanted birth control. They just like they like stick you with a little thing of like haldol, and I don't know where they put it, but I think it's like here somewhere. I think they put it like there. And the haldol is like titrated into your bloodstream. It's an antipsychotic that has a fairly long like half-life anyways. It has like a 50 or 60 hour half-life. Haldol is like one of the go-to antipsychotics that like it's like you're having a serious problem, Haldol, like, because it has a long half-life, it has like a slower onset, I think, and it has like it's pretty widely used, and it's also used in surreptitious medication in the United States, which is crazy. Anyways, so the point is, you take this allergy medication, and it ramps up the production of the enzyme that uh, metabolizes certain antipsychotics like Haldol. And it's not like there's like some chemicals, and there's some like chemicals that activate the cytochrome P450 complex, or that activate. There's some chemicals that activate an enzyme to be able to metabolize a drug, and then there are other chemicals which actually like induce the production of the enzyme. So by taking this like over-the-counter allergy medication, you're basically like causing your body to not only like degrade this drug in particular at a faster rate, it does so in a way that like causes it to always like it causes it to degrade at a faster rate, uh, because it's producing like the cytochrome P450 enzyme that works on this drug. So like taking this triggers the production of this like gene that produces an enzyme that like breaks this down. So it like starts breaking this down faster. The point is like over-the-counter medications, like if they affect like how quickly drugs get broken down in your liver, then that can like completely change like your the chemistry of your like blood. Like that can completely change like whether or not some drug is active or inactive. And basically, it's just really interesting because there's all this talk about predictive medication or predictive medicine, and there's all this talk about like, we have to like read everyone's genome, because as soon as we read the genome, we know exactly how medications are going to work, you know, like perfectly. Like all we have to do is look at a couple like genes on somebody's like genomic report, and like, oh, they've got these alleles of these genes, that means that this medication works in this way, and it has to be like super science because it's science. It's like, no, actually that's not, like, just reading someone's genome is not really gonna like give you that much, because you don't know the state of like their liver, and you don't know the state of like the cytochrome P450 like complexes in the liver that break down all of the active drugs in your body. Like the cytochrome P450 enzymes in your liver break down like 2/3 of the drugs that like are in your bloodstream, and like there's so much complexity to this stuff. It is so impossible to understand in like complete detail. But the point is, like, even if you sequence someone's genome and you know that they have like a particular allele that reacts in a positive way to a particular drug, you don't know like their environmental history that determines how quickly they break down those active chemicals. And that probably has way more to do with the drug's action than like whether or not they have a particular allele of a gene. Um, Although specifics of like the genotypes for like an enzyme or receptor that a drug acts on can be significant, they are, in my opinion, not as significant as the state of like someone's liver and the enzymes that break down the drugs as your blood, streams through your liver. Like the state and quantity of like all of these different kinds of like enzymes, to me, is far more relevant than like some arbitrary like genotypes on someone's like genome for a specific gene that interacts with a drug. And why is that? That's because like this like the liver is always going to be part of the process. Like whether or not you have an allele or whether or not you have a specific genotype that reacts poorly or reacts well to a specific medication, like sometimes that has an effect, sometimes it doesn't. But like the state and quantity and like uh, differentiation of cytochrome P450 enzymes in someone's liver actually has a much larger effect. I would think. I don't know, because it's always that effect is always going to be processed. Like, so, anyways, this is basically my channel right now. I might be streaming on livecoding.tv. I wish I could. I feel like I could get away with streaming on livecoding.tv, but yeah, I think I am. Problem is, I'm like blogging, but I am blogging about programming because I have this like theory about genomics in general. And yes, metanomics is a thing. It is named like perfectly appropriately, as it should be. And metanomics is like the study of like genomics, epigenomics, and then you've got proteomics or something like the proteome kind of like specifies like the state of like proteins that have been produced in a cell or in someone's body. I think there's like several layers to everything. There's like the genomic layer, which is just the raw DNA. There's the epigenomic layer, which includes markings that are laid down on the DNA, and then there's the proteomic layer. Anyway, so this metanomics like extends from genetics to this like theory that I've been thinking about called probabilistic programming, or it's like probabilistic programming as applied to genomics and metanomics. And probabilistic programming has a lot to do with, it's like a useful technique for artificial intelligence and other things. Although, like the sense in which it is like I might change the name to something like, or like it has a lot to do with ergotics. And I don't actually know enough about ergotics to like adequately state how it relates to ergotics. Or ergotics, whatever it is. Ergotic theory. Ergodic theory is like, there's a really great picture here. Like you can look at all this stuff and go blind all day long. It's a really good picture. This explains ergotics. Basically, like you got this system, and as long as like things are like don't get way too out of order, the system like will distribute its like possible states across this like region of like space. Of course, this is a region of space that is like momentum and position. Now here's another one with energy and position. But you can see that like you can track the specifics of this function, but eventually it like blurs down to like this thing. And the reason why it blurs there is because it finds some minimum, or it finds some like efficient like space to occupy that allows like the function to like harmonize uh, and like stabilize. And basically, like ergodics has something to do with like determining like all the like interlinked states or something. Like there's like some particular ergodic hypothesis or something where it says like the probability that these two like red and green dots are like at any particular state must be like the same or something. Like it must converge to some like equivalence or equidistribution, I think is what it is, or something. And so that's what I'm saying. Like I don't actually know enough about this math, but I know that like, so this right here is like a physical system, and it's talking about particles that have momentum and position, and this is what it looks like here, and this is what it looks like in terms of like energy and position instead of momentum and position. And like, see, if the function was slightly different, like this one of these molecules could like pour, or one of these particles could like pour over into here, and it would like find, it might converge at another like uh, equidistribution or whatever. It might converge or it might not. I don't know. Point is, this could be a physical system, or this could be some kind of like probabilistic system. And what I'm saying, my big shtick is basically like metanomics combined with probabilistic programming provides a model for like, or provides a theoretical framework for modeling like any kind of like genomic like it models like biology from like DNA all the way up, and it models it in such a way that like crosses over into programming. And the point, like the other distinguishing feature that I talk about is like spatially distributed state. And it's, what I mean is like the protein products and like the like chemicals produced by like cells and stuff in your body they're like variables in a computer program. Like you can think of those protein products like that are created after a gene is expressed as like variables in a computer program. And basically like the computer program's behavior changes slightly over time. And what I'm trying to do is link that concept, the spatially distributed state, with this ergodic theory over here, and explain it in such a way that makes sense and like doesn't make me sound like a complete idiot when I talk about ergodic theory and I have no idea what I'm talking about. But basically, instead of like modeling like a physical system with momentum and position, I'm going to be modeling like a state machine with spatially distributed state. So anyway, that's what I was going to write about in just a little bit. It's actually kind of a revolutionary idea because I've heard this idea in bits and pieces. This Quantum magazine was like similar, but I mean, like I've never heard anybody talk about like everything from like the top down. Like I've never like heard anybody like link like genomics, epigenomics, like metanomics. I mean, like metanomics is the closest thing to this, but metanomics stops short of saying like this is a program. And what I'm saying is like no, like biology and life is like a program where there's no lines of code. Like all it is like random chance, like random events, and like in spatially distributed state. So instead of variables, you have like proteins and products being created, and just over time, like higher order behavior emerges. And yeah, anyway, I mean, like, I don't know, I'm not really trying, like, I wish somebody would be like, no, dude, that's pretty much the same idea as like this over here. But like, no, like, I can't find anyone who has like most of the time when I talk to this about people, they don't know what to say. Like nobody knows what, like, probabilistic programming is kind of like, it's kind of an older concept. It's not entirely new, but it's kind of something that's on the bleeding edge of like artificial intelligence. Like probabilistic programming itself, just by itself, that's on the bleeding edge of artificial intelligence. Metanomics, that's on the bleeding edge of genomics and epigenomics. So I would, in order to like discuss this with someone, I would have to find someone who like understands both. And I can explain it to anyone who understands like the basics. I can explain it to them. It's just there's not very many people that I find who want to like sit there for like 30 minutes and listen to me talk at them about metanomics and like probabilistic programming. I can't, like, it's really hard to find anyone who would like want to sit there and listen to me talk to them about it. I find it incredibly interesting, and I've got like, I mean, this is one of like my larger ideas. And probably like I could write like 50,000 words on this easy. I'm gonna try and like rein it in at like 20,000. But these are powerful ideas that like, and I'm trying to like figure out like what is the value proposition. Like, how do I relate this to someone who doesn't know what either metanomics is or probabilistic programming? Like, how do I relate this to someone? You see what I'm saying? Like, there's like a crossover for like the ergodic theory to me is like kind of a new thing. But that just basically means that like it's not exactly ergodic theory because it's not. It never reaches like it never reaches like a balance point. Like it never reaches like complete equilibrium between states, which is required. Which is odd because you can never really like precisely define the boundaries of a function that achieves ergodic equivalence anyways. So how do you say that, like, because you can like look at like, oh shit, you can observe in the picture here, you can see like the boundaries of the values, like the boundaries of like the domain or whatever, change significantly. There is like, you can see a clearly defined boundary, but at the same time, like in reality, that's not so clearly defined. It's like very blurry. And so how do you say that some state has been has been distributed around a space? Like how do you say that like some like state has been like distributed enough to say that yes, there is a convergence or not? I don't know, but it's really interesting. And the difference, what I'm saying is like, you could have like one set, like you could have like ergodic transformations. So like this function could change, and it would still be a function like very much like this, where there's lots of random behavior. But because like the underlying function changes, that means that uh, it's possible for this thing to go through several different transformations, and it's possible for this to for a system like this to exhibit like uh, list-like behavior and tree-like behavior. Like it's possible for it to have tree-like transformations, and it's possible for it to have linked list-like transformations. And basically, like if you have something that has like trees, it has like trees, it has lists, and it has like higher order like abstract syntax type stuff, which is basically like if you have any system that has like trees and lists, and really instead of just trees, it needs like trees of trees and lists, then you got programs. And if it has trees, then it has lists. So basically, the only thing you need for like programmatic behavior or like programs really or software is trees. And see, that to me, that's what is so radical. And like, I kind of want to distance myself from the artificial intelligence uh, part of this, because like I want to so probabilistic programming like picks up some baggage, and if people associate it with the same probabilistic programming that is in artificial intelligence, they're probably going to have the wrong idea. And it's not really probabilistic programming. It kind of is, but it's like it's hard to explain, but it's like the most beautiful insight into like how biology works, and I've been like holding on to it for a year, and like I'll tell people about it occasionally, and they're like, "Oh wow, cool," and I'm like, "Yeah, like I'm gonna write about it. I'll write like 20,000 words that no one will ever read," and they're like, "Oh wow, that's cool," but like it kind of stops there because they don't really get the whole. I get the feeling that they don't really get the whole idea. I have talked to a few people who like actually do get it, but it's like it explains like biology as programming. Like I always knew that like, oh, DNA, that's a program. Like, duh. Like it's encoded instructions. But I'm like, there's like so many missing pieces there. I'm like, how is it a program? It doesn't have a defined starting point. It doesn't have a defined stopping point. It can't control like what is really going on. Everything has to be autonomous. This like solves all of those problems. It's like, I don't know. I need a better analogy. See, I'm not really ready to write this, but I kind of want to write it. And that's what, like, oh, I really need to be in, like, Berkeley or, like, San Francisco. Really, really, really need to be in a place where, like, I can just walk down the street and, like, I can yell, like, names of programming languages and people will look up and then I can be like, oh, that guy. And then I can go, like, make a friend. That's San Francisco. You can just yell out names of programming languages, and then you can make friends that way. That's how easy it is in San Francisco. If you try that in Salem, Virginia, people will like look at you like you're crazy, and that's fair. That's fair. I would actually totally do that. I would probably like if I moved to San Francisco, I'll just like walk down the street and be like, "Closure, Ada, fourth, brainfuck, uh, Jade," which is not a programming language. I'm sure it is, though. It probably is. Okay, I'm going to stop recording. Because I didn't even intend to record anyways until I got back from doing the thing that I can't say